Helen is a senior lecturer at Sheffield Institute for Nucleic Acids in the UK. And she's actually very famous for the trainees in your thesis, or we always put this famous Nature paper for synthetic lethality back in, I think, 2005. You can correct me.
No, that's right. 2000.
Yeah, Helen. So it's Helen Bryant.
A young postdoc, I should say. I'm not as old as that.
So the floor is yours.
Okay, so can you see my full screens and my slides and everything? Yeah. Okay. So today I'm going to tell you a really early story, and there's not actually very much mechanism to tell you. It's quite observational, and it reminds me a little bit about when I first talked about PARP inhibitors, I guess, as a postdoc. So let's start the talk. So the story starts with the idea that two pathways are synthetic lethal and that these can be exploited in cancer therapy. And this is well known now. So there's two pathways, there's PARP and there's BRCA2, and you can use either for the cells to survive. So you can inhibit PARP or you can delete BRCA in a tumor cell and everything's okay. But if you get rid of both of them, the cells die. And the consequence of this is that BRCA-like cells will be sensitive to PARP inhibitors, while the surrounding tissue will not be sensitive. And so hopefully what we have is a more specific and hopefully kinder therapy. And here we have the first in vivo demonstration from 2005, where BRCA mutants and wild-type xenografts were treated with PARP inhibitors, and only the BRCA xenografts treated with PARP inhibitor regressed. And years of work followed by many scientists all around the world, and all that work together contributed to the FDA approval of the first PARP inhibitor for BRCA-associated ovarian cancers in 2014. But today I'm not going to talk about PARP, I'm going to talk about PARG. So what is PARG? Well, when PARP takes NAD and adds ADP ribose subunits to acceptor proteins, this is reversed by PARG, or poly-ADP ribose glycohydrolase. And all but the final, the terminal ADP ribose is removed and the NAD is recycled. So the title of my talk is Similarities and Differences with PARP. And it's easy to see how similarities might occur as they're steps in the same process, but why might there be differences? Well, the first reason is perhaps it's wrong to assume that the lack of ADP ribose on acceptor proteins will have the same consequence as excessive ADP or fixed ADP ribosylation. And I think this can be illustrated by looking at one of the functions of PARP. That's its function in single-strand break repair. So following induction of a single-strand break in DNA, PARP binds to the DNA, activates and auto-ribosylates itself. This allows the repair factors to come to the site. And then following attraction of the downstream repair factors, PARP is displaced and repair continues. PARG functions to remove PARP from the bound and unbound PARP, and we can maybe discuss that. And when PARP is inhibited, so when PARP is inhibited, it doesn't have any catalytic activity. It's bound and trapped on the DNA, and downstream single-strand break repair is reduced. When PARG is inhibited, single-strand break repair is also reduced, but to what degree trapping occurs has not been very well established. Equally, the effect of having this large modification trapped within chromatin is not clear. And likewise, it's likely that by fixing the ribosylation, you'll deplete cells of NAD and therefore starve them of that source of energy. And what we can see here in single-strand break repair is likely to be true for other PARP-driven pathways, for example, its function at replication forks, and may also be true for the lack of reversal of other PAR-ated proteins. And the second reason why there might be a difference is that PARG will prevent the reversal of poly-ADP ribosylation by other PARPs other than PARP 1 and 2. So when most people think about PARP, what they're actually thinking about is PARPs 1, 2, and possibly PARP 3. But actually PARP is a large family of enzymes, 5 of which catalyze poly-ADP ribosylation. We've got PARPs 1 to 3, which are DNA activated and play prominent roles in DNA repair, as well as some other processes. And then tankerases that regulate a variety of other cellular processes, Wnt signaling, telomere maintenance, mitosis regulation. And these tankerases aren't also ribosylated, but they interact with target proteins and regulate their interactions and stability via ADP ribosylation. So why does this matter? But when we refer to PARP inhibitors, we're mostly thinking about inhibition of PARP 1, 2, and to a lesser extent, PARP 3. But when we talk about PARG inhibition, we're likely preventing deparation by a much larger group of PARP enzymes, and thus the range of functions that PARG inhibitors may impact is greater. So bearing all this in mind, we considered 2 possibilities, initially looking at PARG synthetic lethal interactions, and then looking at PARG inhibitors to sensitize to DNA damaging agents. And when we started this project in 2010, there weren't any cell permeable selective PARG inhibitors. So we started quite small with PARG siRNA and the non-specific crude inhibitor galantamine. But we did manage to demonstrate that, like PARG is synthetic lethal with BRCA2. Later, working with some chemists in Manchester, we confirmed this with the first-in-class specific PARG inhibitor, PDD17273, or PD as we called it. And this gave us a starting point to screen which other repair genes might be synthetic lethal with PARG. So we screened a library of known DNA repair genes for synthetic lethality, and there were common ones and discrete ones. Here we have an example of some of the ones that were common to both. And then there were those that were synthetic lethal only with PARG or only with PARP. We validated these and followed them up and did some dose responses, etc. And it wasn't just us that found this difference. 2 years later, the Shen and Yu lab screened a panel of breast cancer cell lines using a different PARG inhibitor, the COH34 molecule, and they saw differences in sensitivity to PARP and PARG inhibitors as well. So you can see here, there are just some cell lines that are different. And tantalizingly, when we tested BRCA2 revertants that were no longer sensitive to PARP inhibitors, so here we've got the CAPAN cell lines that are exquisitely sensitive to PARP inhibitors, we've got the wild type cell line, and we've got some revertants, some BRCA revertants that have restored the last BRCA motif in allowing interactions with RAD51. These have no sensitivity to PARP inhibitor, but when we tested sensitivity to PARG inhibitor, they did have some sensitivity to PARG inhibitors. And Shen and Yu also saw this, but in ovarian cancer cell lines. So why is it? Well, we've looked at lots of possible reasons, and we're only just beginning to understand this, I think. But we do think it's got something to do with replication fork dynamics. And you can see that a little bit here in this relative sensitivity to PARP and PARG inhibitors in cells that are overexpressing cyclin E. So when cyclin E is overexpressed, the cells are slightly more sensitive to PARP inhibitors. Here we've used olaparib at 1 micromolar, but they're a lot more sensitive to PARG inhibition. You can also see this if you line up the GI50 values from a panel of breast cancer cell lines with cyclin E expression. And there's a correlation between those 2 factors. And consistent with this finding, a paper from Steve Taylor's lab reports that they see acute sensitivity in cells that are deficient in factors that increase replication stress. So I wish I had an answer for you of exactly what's going on here, but we do have a sort of working model in the lab that we're working towards. And this is how it goes. When certain types of replication stress occur, particularly without overt DNA damage, such as expression of an oncogene, the replication fork can reverse. And we know that PARP can bind to this and be activated at it. And then once the repair has occurred or the situation changes, PARG can reverse the activity of PARP, let's go back, and the replication fork can continue. And at least at telomeres, we know that the molecule RECQ1 has a role in this, and that PARP can ribosylate RECQ1, preventing it from reversing the replication fork. So if PARP's inhibited, RECQ1 could reset the fork without repair. PARP would be trapped, but we don't know that this would happen at a normal replication fork, but this is our model for it, and that the fork could be reset without repair. But if PARG is inhibited, RECQ5 may be fixed off, and therefore restart may not be possible. Of course, there are other models that could be proposed. So we've looked at synthetic lethality. What about sensitization? And here we looked at radiosensitization. The first thing to note is PARP is activated after ionizing radiation, and that PARG increases and prolongs this activity. And we saw that both PARP and PARG inhibitors radiosensitize, as do siRNA against both of these genes. So far, so good. But then we had a surprise. So after ionizing radiation, DNA repair can be monitored. The kinetics can be monitored by looking at the resolution of H2AX foci. And as other people do, we see that at around 6 hours, there's a delay in H2AX foci resolution with PARP inhibition. But surprisingly, PARG inhibition seemed to speed up the repair of DNA damage. And to understand this, we tried looking at downstream markers of repair. So RAD51 foci behaved similarly between PARP and PARG. But when we looked at DNA PK foci, what we saw was a massive activation in both the number of cells with foci and foci per cell after PARG inhibition. And this suggests that PARG inhibitors can increase the recruitment of non-homologous end joining factors to DNA damage. In addition, we looked at what mitosis looks like after ionizing radiation, and we noticed that PARG inhibitors caused a significant increase in mitotic aberrance and accumulation of the cells in metaphase. So what could be going on here? Normally, we understand that PARP1 can promote resection at double-strand breaks and therefore homologous recombination, and in a way suppress non-homologous end joining. PARP3 is capable of ribosylating Ku to promote non-homologous end joining. And there's one paper that reports that tankerases can be recruited via MDC1 to promote the stability of BRCA1 complex and therefore contribute to homologous recombination and contribute to checkpoint activation. And through these different activities, the different PARP enzymes contribute to a balanced homologous recombination, non-homologous end joining outcome after radiation. If PARP1 and 2 is inhibited, we know that there's a lack of single-strand break repair, that PARP becomes trapped at forks, and that PARP activity is unable to stabilize these forks. And it's likely that all of these contribute to increased DNA damage and delayed repair via homologous recombination. PARP3 is likely to no longer pyrate Ku, but the action of the tankerase would be unaffected. However, when PARP's inhibited, modified PARP is bound on DNA, and we think perhaps the forks still collapse and might need homologous recombination for repair, but we don't know that. PARG likely still modifies Ku, and this stays pyrated, and maybe this could be promoting non-homologous end joining. And then the lack of reversal of the tankerase activity might alter the stability of the BRCA1 complex, and checkpoint activation might be altered. Okay, school's just come out. In addition, both PARP3 and tankerase have function in mitosis, where activity is increased. By activity acts to help form spindles. So when PARG is inhibited, we can postulate that this will alter the spindle dynamics. So you can see how both altered functions for unmodified and trapped modified PARP could have different effects, and how changes in the acceptor proteins could be different, and also how lack of reversal of tankerases could contribute to the phenotypes that we're seeing. And consistent with this, we see that tankerase inhibition also radiosensitizes and causes mitotic aberrance. So in summary, we've seen that PARP and PARG inhibitors share some therapeutic vulnerabilities, but also that each has a unique set. And that PARG, and we suggest that PARG inhibitors in the future will offer distinct therapeutic opportunities compared to PARP inhibitors. It's the very beginning of the story, in my opinion, and the understanding of this mechanism is essential for us to exploit it further. And also, the drugs that are available at the moment are quite early stage and certainly need a lot more development before they can be used as clinical agents. So I'll thank the people who did this work. So most of it was funded by Breast Cancer Now or Yorkshire Cancer Research and was carried out by a postdoc, Polly, and a PhD student, Emma. And then the chemistry of the PD compounds and our collaboration with them was through James in Manchester,